to Dr. Jariwala, and here's the first one. Is it possible to include a manufacturer of an intermediate which has not been audited by FDA in a DMF?
Yeah, that's a great question. Excuse me. Yeah, you can definitely include that in DMF. If that facility also manufactures API, more than likely, if they are new registrant with us, more than likely they will be inspected as part of any subsequent NDA or NDA that they are named in. But we do, I mean, there is no limitation why that can happen.
Thank you for responding to that question. The next question, when will US FDA resume foreign facility inspections which have been on hold due to the pandemic?
Again, a great question. And I think there are a lot of questions along the lines. I'll just take a little bit higher level view of this. As many of you know, we are currently conducting inspections of that we deem mission critical. There are many factors that go into this. So what I highly recommend that if your facility is OAI or for any of the reason you believe, we recommend that you communicate with your point of contact, whether if it is application related or it is GMP related. You send us your request. You also, if there is any way, for any reason you are making a COVID therapy, drug shortage product, or a breakthrough therapy, mention that. That helps us understand situation better, and we will see what we can do. I do not have a time frame right now, but we are conducting if they are deemed, if we deem them mission critical. What I highly recommend, you keep the lines of communication open, send in those requests, along with any of the reason that may make us think that that inspection should be a mission critical inspection. Some of the things I outlined during my presentation, so I highly recommend that.
Thank you for responding to that question. The next question, does FDA inspect API starting material manufacturers?
Well, the answer is maybe, right? How critical the intermediate or the starting material is. If that facility also makes API, including starting material. So the answer is yes, we do, but it all depends on the situation.
Thank you for responding to that question. The next question, what impact does COVID have on inspection? How much do you accept foreign government inspections?
I don't have to explain the impact. But I think the second part of the question is great. As I talked during my presentation, we accept foreign government inspection that are allowed under the MRA. We collaborate with them as long as they are conducted within their boundaries. And we do have confidentiality agreements with other countries that we can request data from, but that is something we do it on our own. I think that the manufacturer can do in that. So we do accept inspections that fall under the paradigm of MRA.
Thank you for responding to that question. We have a couple more questions for Dr. Jariwala. Does FDA issue an Establishment Inspection Report, or EIR, for a secondary site inspection every time? If yes, what is the timeline for receipt of this post-response submissions on 483s?
Okay. I do not understand what the secondary site inspection, but for every inspection that FDA conducts, at some point we do issue EIR. Typically under CONOPS, we are if it's a GDUFA facility, but I mean we are doing that for non-GDUFA facility as well. That we try to issue a decision within 90 days of the close of the inspection, and if it's an OAI, then a regulatory action within 180 days. So that's the time frame. So if a facility is NAI or VAI, you should expect EIR along with that 90-day decision letter. If a facility is OAI, then it would be about 180 days when we issue a regulatory action. We typically send FMD 145, and along with that, we send EIR.
Thank you for responding to that question. And one more question for Dr. Jariwala, and this is the last one in this section. How should a firm provide documents to FDA?
Another great question. So there are three things I would highly recommend. If during the inspection, when a FDA 483 is issued, if one is issued, there should be an email address identified on that investigator, or just ask the investigator. They should be able to show you. Second, if you are trying to communicate about your facility that is part of an application, then you should always follow whatever the application holder and their point of contact is. And if you are trying to communicate about any regulatory action or the inspection OAI, you can communicate about that depending on if it's a domestic facility, then through our district offices, or if it's a foreign facility, then directly with Office of Manufacturing Quality. And if you go on FDA.gov website, there are email addresses listed for that.
Thank you for responding to that question. The next group of questions is directed to Dr. Connell. And here's the first question. Could filing a DMF amendment delay the approval of a referencing ANDA that is set to receive approval?
Yes, to answer that question, yes. Filing an unsolicited amendment could extend the review timeline. So we recommend that DMF holders keep open lines of communication with their referencing ANDA customers to minimize the type of delay that would be caused by our receipt of an unsolicited amendment submission to a DMF.
Thank you for responding to that question. The next question. Please explain why this guidance does not give reporting categories for all types of changes, only for some types of changes on the last 2 pages, but it didn't give you the actual guidance title.
Yeah, that's a good question. We give recommendations and examples in each reporting categories. There's simply too many variables for us to give a comprehensive list of everything. So it's really a case-specific decision. So we stress the importance of a robust risk assessment when deciding the appropriate reporting category, taking into account all the information that you have available about the process. Thank you.
Thank you for responding to that question. The next question. During new source qualification for starting material, is it necessary to use 3 different SM batches, or would a single batch be acceptable during API validation batches?
There's no strict requirement for 3 batches. Of course, that is often done. If the starting material, for some reason, the batches are much larger than the process batches, that will be the starting material used to make the drug substance. The starting material batches are larger than the process batches. It is possible to justify a change, but of course this would be part of the risk assessment that should be done when proposing to use a smaller number of batches than is typically seen during process validation. Thanks.
Thank you for responding to that question. The next question. If I remember clearly, your out-of-scope slide did not list specifically products of fermentation. It listed semi-synthetic products and biotech products. Does the guidance apply to API produced by classical fermentation?
Thanks for that. That's a good question. So if you go back to slide 4, if you download the PDF and look at slide 4, that lists the out-of-scope types of drug substances. It says non-synthetic steps such as fermentation of semi-synthetic drug substances. So any step that involves fermentation is out of scope. So if the drug substance is a direct product of fermentation, it would be out of scope of this guidance. If fermentation was only part of the process, so a semi-synthetic drug substance, the part of the process that was synthetic would be applicable to this guidance, and the part of the process that was non-synthetic would be out of scope of this guidance. Thanks.
Thank you for responding to that question. The next question. Is the post-approval changes for drug substance guidance not applicable for both synthetic and recombinant peptides?
Yes, that's correct. It's out of scope for any type of peptide drug substance, yes.
Thank you for responding to that question. Got a couple of questions left for Dr. Connell. What are the requirements for choosing pre-change batches to compare to post-change batches? How many batches should we select?
That's a good question, and I would say it's up to the risk assessment, but generally the more the better. If you have lots of pre-change batches that you can use to demonstrate comparability, you know, we've seen submissions with data of hundreds of batches. If you can do it, if you're early on in the you know, life cycle of the drug and you have fewer batches to show the impact of the change, that can be acceptable too. We generally do see 3 batches of post-change material, but if you have more, certainly I'd say that's likely to be better, particularly if the statistics work out better. And if there's less and it's justified by the risk assessment, you know, we take everything into consideration. But again, if you can present your risk assessment for why you think that's the way to go, then we'll take a look at that. Thanks.
Thank you for responding to that question. Looks like we can squeeze in just one more question for Dr. Connell. If the structure of impurity X is unknown, how can we know what will be its derivative Y?
That's a good question, and if you're observing X at a level such that you have observed it as a new impurity, you need to do some analytical chemistry, some characterization of that, maybe isolated mass spectrometry maybe could help, but we would expect some type of effort to identify the structure of that impurity and then get it qualified so that if it's going to be reactive or not reactive in the next step, and if it could possibly generate some impurities that are of a bigger concern, such as mutagenic impurity. So whatever you can do to figure out what's showing up is what you need to do in these cases if you're trying to go for equivalence of the impurity profile. If you're taking that approach, it's important to be thorough with that approach. Thanks.
Thank you for responding to that last question. Well, that's all the time we have for this session. We're going to move into our break right now.